So that's it, really. Yeah, it's great to see everybody, and I really appreciate, well, everybody coming, but the ones that are also sitting up at crazy hours and not at 10 o'clock European time. So thank you very much for your interest. And that's it, really. I think we're going to start the recording and start with the first presentation, which is from Mark Koch from the SeaMark project, and Mark is from Station Biologique de Roscoff. Sorry, dreadful French, and you can do it better, Mark. But
not much.
On you go.
Okay, thank you, Maeve. Can we put up the first slide? Yeah. So I'll be talking about work we're doing in Roscoff at the moment, trying to put in place breeding approaches for Saccharina latissima. Next slide. So as probably most of you know, at the current time, Saccharina culture in Europe and North America relies almost exclusively on locally sourced genotypes. There hasn't been any input of breeding into improving the strains which are used, but I think there's plenty of scope to do that. In the next slide. Yes, for instance, for land plants, over many decades, there have been really enormous improvements in terms of yields and other characteristics. So I think there's a lot of scope that shows that there's a lot of scope for improvement. And actually, if you can just click once more, in China, there's been for many years now a breeding program for Saccharina japonica, and that's made some important advances in producing high yield strains for that species. So I think we can really improve latissima in the Western Hemisphere. So the next slide. So in Roscoff, we're really doing two types of activity. One is sort of generating background resources for Saccharina breeding, and the other is sort of trying to make some first steps towards implementing a breeding program. So I'll go through those two aspects. Just before we go to the next slide, this has involved SeaMark, but also several other projects which are listed at the bottom of the slide, and I'll try to sort of give an indication of which project was involved at each time. So the next slide. So I'll start with the resources, and the next slide. So the first resource was last year. We published a genome assembly for Saccharina latissima. This is part of the FAO Explorer project, where we sequenced about 60 other brown algal genomes. And in the next slide, sorry. Yeah, this genome is freely available. It's in the public. Databases. It's also on a website in Roscoff, a FAO Explorer website, where you can easily access it and download the data, do blast searches in genome browsers, and so on. The next slide. Sorry, I'll go through quickly because I don't have much time. Within the Genealg project, we've also this year published a genetic map for Saccharina based on dd-rad sequencing. I won't go into details of this, but in the next slide, we actually used this genetic map to go back to the Saccharina latissima genome and improve it. And we have a V2 assembly now, which is chromosome scale. This was Perla Farhad that did this work. And this hasn't been published yet, but it's already available. We've made it publicly available on the FAO Explorer website. So next slide. So in parallel, we're trying to develop sort of functional genomics methodologies for Saccharina. As part of Alexander Jetterbock's Kelp Prime project, we're trying to get CRISPR working for Saccharina in Roscoff. And then within SeaMark, there's an activity which is really centralized in Copenhagen at Tritomic, where they're trying to develop a sort of tilling approach to have a large collection of mutants and screen them, which would be a sort of non-GMO equivalent of the CRISPR method, and could be used both for fundamental research and also for potentially for producing improved strains for culture. And then the next slide. And so the sort of final aspect of the resources is we've also established a large collection of clonal gametophytes in Roscoff, nearly 600 strains. from multiple sites right across Europe, and these have all been genotyped using dd-rad sequencing. So with those resources in hand, the next slide, we're starting to sort of make some first movements towards Saccharina breeding. Before I actually talk about breeding itself, I just wanted to mention rapidly, we're not involved in this. This is mostly Alexander Jutterbach in Boda and Inka Bartsch in Bremerhaven. But they're within this kelp prime project. They're working with priming to actually try to apply this to Saccharina culture. So the idea behind this is you during the production process, you apply a little stress to your material. And when a stress reoccurs, for instance, during the cultivation period of the seaweed, those strains will normally be more resistant to that stress. So this is something which doesn't involve actually changing the genetic basis of the organism, but can actually be interesting to improve strains in the short term. So in the next slide, we'll go on to sort of the real breeding activity. And as I said, we've sort of just made a few first steps with this. So Zofia and Francis have been doing quite a large number of crosses within the SeaMark project, and you can see on the right there's quite a lot of variation in between the phenotypes of the F1 sporophytes. So we do have variation, which is interesting for breeding. And another interesting result from this was that we did both intra-population crosses, so individuals from the same site, and also wide crosses between Faro and Brittany, because we wanted to actually sort of validate an intra-population approach so that we could actually use that approach in future for breeding and avoid bringing in foreign genetic information into local sites. And what was nice was that we didn't see any really significant differences in terms of results between these intra and inter crosses. So it looks like this sort of breeding with using just local strains is actually a valid approach. And the next slide. In the genealogy project, we've gone a little bit further than just these F1 lines. So we've actually created an F2 segregating family and looked at growth and also temperature stress tolerance in that family. And I'll just say a little bit about the temperature stress. That's on the next slide. So Zofia did a temperature stress experiment, the actual degrees. So she took leaf blade explants and then treated them to the stress or not, and looked at physiological responses, photosynthesis essentially, and transcriptomics. And what was nice here is that we see, again, variance within this segregating family. There are tolerant and sensitive and resilient individuals. So again, there's material there for breeding. And in the next slide, we were able then to use the genetic map, obviously, to look at, to try and look for QTLs, quantitative trait loci associated with this, with these characteristics. And Zofia detected 3 QTLs related to recovery from stress. So these are quite large intervals on the chromosomes, but so we're not narrowing down to individual genes, but they give us some loci which could be interesting for breeding, and at least some indication where we might be able to find genes that are important for temperature stress. And the next slide. So I just wanted to finish by thanking all the people involved in this. Time is very short, I think, so I'm not going to read through all the list, but you'll see Zofia's name appears quite a bit here in these different aspects. And I've just tried to list the various people involved in the different aspects and who has been coordinating the different projects. And at the bottom, to acknowledge our funding sources. And thank you for your attention.
Thank you, Mark. Would anybody like to ask 1 or 2 questions? We have a couple of minutes after each presentation for that. I think you can just raise your hand or unmute your microphone.
Sorry, I went very quickly, but minutes is not long.
we don't have an awful lot of time, unfortunately. Have you got any oh, sorry, there's there's a hand has been raised. Sarah, go ahead.
You're muted, I think.
Sarah, we can't hear you.
No.
Maya, do we have to unmute everybody?
I think she's
No,
unmuted.
she is unmuted, but I think there's something wrong with the microphone. So maybe we
Okay.
can skip to Gabriel in the meantime.
Hello, can you hear me?
Yes.
Yes.
Thanks. Thank you. Thank you, Mark. I know it's very quickly. I was aware of pretty much all these papers and research of Roscoff. So 2 questions very quickly. I hope so it's not too long answer. You outplant those individual was outcrossing between population in farms already? That's question 1. And the second is what is the capabilities of mass produce these outbreeding or potentially breed lines?
The experiments were all done in tanks in Roscoff, so we haven't released any of those strains from the crosses. So that was we needed to do that because we were doing the white crosses as well, and we didn't want to release that material. So all that has been done in under control conditions. But the objective of that experiment was actually to develop improved strains, and we've only looked at the intra-population crosses for that, and we've picked the best strains, and they've been sent to the Faroe Islands and to Algalesco in Brittany, and they're being bulked up to actually be tested under field conditions. I think that's both questions now. I'm not sure.
And it's fine. The second one is about capacity. So how many meters was produced and how many do you expect to expand?
Well, that's not been done yet, but and we're actually a bit, because we're starting from clonal gametophytes, we're having problems producing enough biomass to really get up to scale to actually do the farming out to the field situation. And we're getting there, and hopefully we'll be able to at least put out a few meters this year, enough to actually be able to do some statistics and test compare these strains with the existing strains.
If you like, Mark, I can give the latest update
Sure.
on the bulking of it. We're back from Ocean Rainforest. So the material or the strains developed by Mark and his team in Roscoff is then bulked up both in Algalesco in France and at Ocean Rainforest in the Faroe Islands. And we have currently like 6 different strains that can be deployed in maybe 5 replicate lines, so around 50 meters for each breed. So it takes a long time. This was in 1 year that we could grow it to this scale.
We have Sarah has written her question, and it's in the chat there, Mark, but I'll just read it out for you. In the F2 family, do you observe any depression in the F2 that can be due to genetic incompatibilities?
So this F2, I skipped over this a little bit, but it was between 2 strains from two parents, one from northern Brittany and the other from southern Brittany. So it's a sort of an intermediate distant cross. It's not from the same population, but they're probably fairly closely related. The northern Brittany and South Brittany populations are actually quite different, but it's not a wide cross. So actually, to answer that question, we would have to compare with other crosses. Miriam Valero has done some work on this, looking at crosses at different distances, and in different experiments there were different results. There was sometimes evidence of heterosis and sometimes evidence of outbreeding depression, but usually you don't see a lot of those effects.
Thank you very much, Mark. I think we have to swiftly move to the next presentation. And this is the Saccharina presentation from the Blue Bio Boost project, and Signe Brottelund is your next presenter.
Okay, thank you, Maid. So yeah, I will be talking about these two breeding experiments with Saccharina latisma. So the first one was a part of the Breed for Kelp to Feed project, actually, which is completed. And in this one, we used mixed hybridization and mass selection and analyzed more than 1,600 genotypes. The second one, which is the one from Blue Bio Boost, has been initiated, but it's not yet completed. Here we use another strategy, which is targeted crosses of single spore gametophyte cultures. And we analyze 48 genotypes in each experiment, and the experiment is done in 3 countries. So next slide, please. First, I'm going to talk about the experiment from Breed for Kelp to Feed, which is completed. With this mixed hybridization and mass selection strategy, we breed for population rather than varieties. So we select a group of parents, we mix them all together in an uncontrolled way, or their spores, and then we get the next generation. It's a relatively simple and cost-efficient method, so it would be suitable for local breeding programs if it achieves genetic progress. Next, thanks. So here you see the scheme from Breed for Kelp to Feed. We started with 159 individuals, mixed all of these, or the ones that's sporulated, together in a mixed hybridization to get the F1. And then from F1, we selected the 40 longest individuals and used 25 of these as parents for F2. So the selection happened between F1 and F2. Next slide. So in a breeding program based on this mixed hybridization and mass selection, the cycles would look something like this. And you see you apply the selection when you select the longest individual from each segment, which is the blue box, but or green. But in addition, you have all of this unintended selection going on, which is the other boxes, because you don't really control what's going on during the mixed hybridization. Next slide. So in our experiment, we did parentage assignment to kind of look at what was going on. And in these bar plots, you see the bars as parents, and then the height of the bar is the number of offspring they have. So from F0, the wild ones, to F1, we saw that there was one parent that had a lot of offspring. And in general, there is quite uneven genetic transmission, with some parents not having offspring at all. In F2, it was more even, but still there are some parents that had very few offspring. We also saw that there is some self-fertilization or selfing going on. So in F2, this was actually more than what would be expected with random mating, which is a little bit strange because we expect there to be some inbreeding depression. We also see that crosses between siblings are rarer than at random chance, but we think it has to do with the methodology when we mixed it together. In F1, we also saw some apomixis, which is the yellow part of the bars. It's just a few individuals, but these were completely homozygous, so they were like super inbred. We also saw very few, but we saw triploids in both generations, which is quite interesting. So next slide. We did some quantitative genetic analysis of this study, where we tried to separate between the effects that determined the phenotype. So we had additive genetic effect. We also had some fixed effects involving spatial effects and whether the individuals were inbred or not, and also an error. We tried to estimate genotype by year interactions, but we couldn't do it because there was not enough of a genetic link between the generations, unfortunately. Next. Oh, okay, so there's some formatting issues there, but you see the results, I hope. So the blade length, blade width, and stipe length was significantly heritable in both years because we had to do it separately for each year, the calculations. Fresh weight, on the other hand, we were not able to determine that if it was heritable because it had a very large residual effect or error. And I think this is because we don't really have genetic replicates in this experiment. The prediction accuracy was decent, I think. One interesting thing is the inbreeding depression, which was significant for all of the traits in both years and quite strong, especially in the 2020 model where these apomictic individuals were. Next slide. Yeah, we also looked at some correlations between traits. Here, the below diagonal is the phenotypic correlations and above is genetic correlations. So we see, for example, that blade length and fresh weight is highly genetically correlated, meaning that if we select for blade length, we will probably improve fresh weight as well. Next slide. To see if we had some genetic progress, we looked at the estimated breeding values, which is like the genetic merit for the trait. So higher breeding values means that they are more genetically prone to grow longer. And if this selection would have worked, then we would expect the breeding values in F2 to be higher than in F1. But if you look at the 2020 model, we see that the light green box, which is F2, and the yellow F1 box is the same. So there was no change in breeding values actually, which is a little bit surprising considering that blade length is heritable and we applied very strong selection. But we think it has to do with what we see in the brown box, which is the longest individuals from F1 that were selected. They don't really have any good genetics for the traits. So it turns out these are probably some kind of outliers that are long due to some random genetic effect, random non-genetic effect or a genetic effect we couldn't estimate. So that's a little bit unfortunate. Another interesting thing is that we see that the yellow the breeding values have actually decreased from F0 to F1 significantly, although we did not apply any selection. So this is the unintended selection that negatively affected blade length breeding values. We also see differences between F2 in the 2020 and 21 model, which means that there is probably G by E interactions, although we couldn't estimate those. Next slide. So for the next experiment, we use this other technique of targeted gametophyte crosses. So what we do is that we take some spore phytes, we isolate single spores from them, and these develop into these gametophyte cultures that have identical genotypes. And then when we cross these gametophyte cultures, we get a group of spore phytes which are also genetically identical. And I see we cannot see the crosses in the picture, but it's not so important. I think something had happened to the presentation. Anyways, since we have these genetically identical individuals, we get genetic replicates, which is really good for the statistics and a big improvement from the last experiment. Since we control the crosses, we also mitigate this selfing and much of the unintended selection. We can also use these targeted crosses to utilize heterosis, which is the opposite of inbreeding depression, meaning that offspring with genetically different parents tend to perform good. And if we get heterosis, we can improve the genetics in only one generation, which would make it a very quick technique to produce cultivating material. Also, we measure the phenotypes on the sporophyte level, but we get actual breeding values for the gametophytes. So this is really nice because then we will know which gametophyte cultures have good genes for the trait, and we can keep those for many years and use them to make crosses. Okay, next slide. So the experiment looks like this. We start with 24 individuals, sporophytes, and from each of those, 4 single spore gametophyte cultures were made. Then these were crossed in a strategic way to make F1, which was cultivated on ropes. Then when we find the best crosses in F1, these will be remade in the next year to validate whether they are good in different environments. So this scheme is repeated in 3 countries. It's Norway, Sweden, Ireland, and in each country use local populations. Next slide. So when we will do the genetic analysis, we think it will be better than in the last experiment. One thing is that we can not only study individual phenotypes, but also segment level traits, since we have genetically identical individuals on the segments. So for example, we can look at yield per meter, which is very interesting. Again, we will estimate the additive genetic effect, but this time we will also estimate the dominance genetic effect, which is very important for heterosis. We will again try to estimate genotype by year interactions, but this time I think we can do it because we have replicates across year. And also we will have probably better estimations of spatial effects and less residual error. So a click. Can you, okay, yeah. So yeah, this is obviously not correctly formatted, but I have not gotten the data yet, but I have run some simulations based on genetic data from the last experiment and this scheme. And it is very promising when it comes to estimation of dominance effects and also for better precision. I haven't been able to test the rest yet. Okay, next slide. So in Blue Bio Boost, we have this mix to match strategy to reduce the impact of genetic progression on wild populations. So the idea is that you collect the wild population and you phenotype it to, yeah, these graphs on the bottom line is the allele frequencies of the alleles. So you know the allele frequencies. And then when you have identified some superior crosses, you also genotype these. And then you combine certain amounts of each cross to make a cultivation population that has an allele frequency that is as similar as possible to the native population. Okay, next slide. So just a little bit about what we have done so far in this project. So we have made the crosses in all countries now for F1, and we will sample those next year. So looking forward to that. This has been very hard because growing single spore gametophyte cultures is a lot of work and it takes a lot of time. Going from one single spore to the amount of gametophyte biomass that is needed to make crosses that can cover this length of rope that we need. We also need to keep more cultures that we will use because in the beginning it's hard to tell if they're male or female. There are also some cultures that just die or get infected, or we have also seen that some of them undergo parthenogenesis, which is that they produce sporophytes from females without fertilization, and then these can't be used either. So that's all I had. Just looking forward to get some data to analyze. Thank you.
Thank you very much, Signe, and sorry for the few formatting issues there on the presentation. You did very well.
It's okay.
Okay, I think we have time for a couple of questions. I'll just go over to the chat, or if people want to put their hand up, that's. Let's see. Peter has said in the chat a recap question. In the first experiment, the F0 generation, you'd select the longest blades, and out of those, about 150 blades, there was only a single parent producing a very high number of offspring. And in this F1 generation, did you see an inbreeding depression effect on length? And if so, why is that?
Okay, so the F0 population was wild individuals, and when those were collected, we tried to not do any selection at all, to just collect random individuals. Yes, but then in F1 we saw that one of these wild individuals produced a lot of offspring, and because of that, there was quite a few individuals in F2 that were selfings by chance from this parent, but it wasn't it didn't result in a lot of inbreeding depression in that generation. I think if we had used a smaller number of parents and some of them produced so many offspring, then we would have bigger problems with inbreeding depression. But when we did the statistic, we saw that the inbred individuals are much shorter than the non-inbred. I think it was 30% on average. I hope that answered everything.
Yes, thank you.
I think we have, is it Sana first there? And we really, yeah.
Thank you. Interesting presentation. I also have a question about the first part you talked about, because you say you selected the longest blades. But you, of course, have also competition between the blades, because I think multiple people showed that there's a correlation between blade length and yield, and so you would expect maybe also a higher yield. Don't you think that maybe because of this density effects, etc., it's not possible to select in this way?
Yeah, I think that is a very important point, because we have seen this technique being used very successfully in China, but there they tie each individual sporophyte with even spacing to the ropes, and we don't have that. So I think there is a lot of density effects that kind of interfere in this type of design, and that is why the longest individuals did not necessarily have very good genetics.
Mark, I think we might have your question, if it's a quick question, and then I think we have to move on. But Pierrick, if you can put your question in the chat, maybe we can get back to it later. Mark?
Let Pierrick go ahead. Let Pierrick go ahead. I don't have a very important question.
Okay.
No, it's not. I don't think it's really important, but thank you, Mark. It's come back to the last question. The goal is to produce algae and biomass, and heritability of fresh weight is close to zero. You partly give answer to the subject, to the question, but how is it possible to get heritability close to zero for fresh weight? And even if it's highly correlated with some other traits, meaning when you have normally high correlation, it means you have additive genetic variation, but you are not allowed to see it. Is that linked to what you say about the methods of evaluation with lower density, which could be requested?
Yeah, I think that fresh weight is definitely heritable. We were just not able to see it in this kind of design. And I think it's because of these density effects, and we don't have any genetic replicates. So it's really hard to differentiate between the genetic and the environmental effects to length. And considering that it's of course intuitive that it would be related to blade length, because if you have longer individuals, they will be larger in general. But I really think it is heritable, and with the design we have in the next experiment, I think we will be able to confirm it. Yeah.
Mm-hmm. Thank you.
Okay. So thank you very much. The sugar kelp presentations, we're going to change now slightly, and we are going to go and talk about ulva. I suppose a different species and perhaps some different strategies. And our first talk is by Inge Hellige from the University of Gothenburg, and I shall leave the floor to you, Inge.
Thank you. Yes, I'm looking forward to talk about the selection of morphologically stable, fast-growing Ulva-Finistrata strains. Yes. So first of all, why Ulva-Finistrata? It's like a key species for aquaculture in Scandinavia. Next. Because it's local, but also produces high biomass. So you have like high growth rates, rapid proliferation. And also it has a nutritional value, so for the food, but also feed industry, and but also cosmetic or pharmaceutical sector. So all of this makes it attractive for large-scale cultivation. Next, please. Oh, there's also some formatting, but that's okay. So if we look at the distribution of Ulva fenestrata in Scandinavia, so on the left, we sampled Ulva species around the Swedish, Norwegian, and Danish coast. And as I already said, Ulva fenestrata is quite local. So here on the right, the red area, so at the Swedish coast, but also southern Norway and northern Denmark. Next, please. So I think it's very important in the beginning to now look at the life cycle of Ulva fenestrata. So we start with our sporophyte, which produces zoospores, which then grow into gametophytes. And these gametophytes again can release gametes, and the mating gametes then develop into a zygote, which then again develops into a sporophyte. Interestingly, the sporophyte as well as the gametophytes look alike, so they are isomorphic. And also, if they're like in absence of a mating gamete, the gametophytes or the gametes can then develop into a new tally without fertilization. So we have this clonal pathway, which we will focus on now. Next, please. So what has already been shown for Ulva compressa is like the development of these slender-like mutants. So what you can see here is an A, the wild type of Ulva compressa, and then in B and C, an overview of like the mature gametanglia, and then in C, after it released the gametes. So they used this for cultivating Ulva compressa, and in D you can see the Ulva after 3 weeks of cultivation, and you see this one slender-like mutant, which you can see again in F, so which is supposed to grow faster than the wild type. So next, so yeah, you can click again, next. So what we are trying to do now is like to develop strategies to enhance the morphologically stability in Ulva fenestrata and also to enhance its growth. So we are trying to select for the fast growing, like these giant phenotypes, like the slender one, and then investigate whether this trait is random or if we can link it, for example, to polyploidy. Next, please. So what we did, we sampled Ulva fenestrata along the Swedish west coast. So if we zoom in here, one more, please. Yeah, we are located at Tjärnö in the Costa Archipelago in the north, and then we sampled around the west coast until a little bit north of Gothenburg, so Marstrand. Next. So in total, we sampled 46 individuals. Of these, 36 were wild living, 2 long-term cultivated strains, and then 8 from Nordic Sea Farm. Next. Next. So what we did, we cultivated Ulva fenestrata in these jars, and once they turned fertile by this color change, we took the fertile material and they released the swarmers, which we then plated into an F1 generation. And after 4 weeks, we analyzed the length of each of the individuals. And then after 6 weeks, we transferred like the giant individuals into separate cultivation flasks, but also took some of the non-giant ones. And then we repeated this screening in the next generations. Next. So here I brought an example of one of the plates. And if we focus now, next please, on this white square, next, then we have this one individual, which is quite large, and then next to it, next, like one of them, which is definitely smaller, but then it's also the question if the first one is at least twice or even bigger than the average of the others. Next. So what we did here, we analyzed the length in millimeter for all the individuals in the F1 generation. And I would like to focus first on the upper one, so the BBB1. And the first line, the dotted line, shows like the average length of like all of the individuals which grew into an F1 generation. Next. And then the second line shows like the individuals which are at least twice the average size. So these were considered like the giant ones or the bigger individuals. Next. So this is definitely one which is like the upper one, which is larger than the others. So we did this for all individuals which grew into an F1 generation. Next. And for example, here you can see on the BBB16 one individual, which is definitely bigger. Next. But also for others like BB14, 15, or 29, where we have quite some diversity in lengths, but still like some which seem to be more gigantic than others. But we also, next, sorry, see some individuals here on top where only like a couple individuals developed. So sometimes only one individual grew into a next generation. Next. And then also 33 and 34 in the middle bottom half, we see like no individuals really growing to a gigantic or larger scale. They are all quite similar sized. So in total, around 61% developed into an F1 generation, so 28 strains grew. Next, please. So yeah, now we are here. We selected the fast-growing giant individuals and transferred them in the bottle. Next. So this is our F1 generation now, and we so we waited for them to reach fertility, to then plate again the swarmers, measure their lengths. Next, and then also again select them. Yeah, next slide. So now we are moved forward to the F2, and here only 12 strains developed into, like referring to the parental origin, still developed further into an F2 generation. Yeah, you can go further, it's okay. So if we zoom into these strains, here again we see the length in millimeter. And in the top row, we now see the BBB1, so every individual which referred like from the first parental individual. And what's quite astonishing is that like in the second generation, they are all relatively small. So there doesn't seem to be huge giant individuals anymore. Also for the BBB3, they are quite similar. But then for the ones on the right now, we see that we still have quite a lot of individuals developing, and then also still find the giants also for the one in the furthest right, where I will come, where we'll talk about a bit later. But also for the other ones, sometimes they are quite small. And then now next, please. But we have some individuals like this one, where all of them, like the length is very diverse, but also we have a lot of huge individuals. So, but it's not like they are not all big, but they are just like very diverse in length. And next. And now I just want to highlight once more, like for one in the middle, like this BBB34, we did not have any giants in the beginning, and still also in the F2, we don't see any giants. But then in the last one with the arrow, where we had like, we picked a control, so not a gigantic one, but then again, in like the F2 generation, we also see one individual, which seems to be bigger than the others. Next. So if we look at them phenotypically, we of course have the wild type, and all of our morphological classification is based on like previously described mutants from Ulva, but we also see in culture like this bubble shape or like a forked individual. We also have this slender type, which was already described as being one of the faster growing ones, or also like a lot of these wrinkled or constricted or even mixes out of them, like out of the two. And what I've seen a lot is also this branch star. So this is defined as like having one center, where then the ulva is growing into like various directions, like derived from this center star kind of. So what we are currently doing is we are cultivating our F2 generation to again reach fertility, to then release swarmers and again measure their length. So this is ongoing work, but for one of the individuals, this BBB8, we already have generated an F3 generation, and it all, it's from one individual in the beginning, which developed into an F1, and then the F2 and F3, has a quite like more diverse offspring, and we also see that the mean length is slightly increasing over time. But of course, also because of one of the individuals in the F3 being more than 10 centimeters big after 4 weeks, which is really big. And I brought a photo. Next, please. Yes, of the one which grew to like nearly or over 10 centimeters after 4 weeks. So what we are planning to do now is we are selecting these giant individual strains for upscaling of biomass, or also trying to link this together with lean Leos from Ghent to determine their ploidy levels. And then, of course, also collaborate with the seaweed industry to see how we can use this selection process, so how we can use these strains in future outbreeding trials. Yes, so with this, I would like to acknowledge, of course, Sophie Steinhagen for all of her help, and a lot of people, helping on sampling or strain, or getting the strains, or like then the ploidy measurements, and all of this is part of the Blue Bio Boost project. Thank you.
Thank you very much, Inge. As usual, it shows an awful lot of work that goes into all of these cultivation projects. Does anybody have any questions for Inge? Not sure if I see any hands just yet. Ronald, go ahead. You're muted, Ronald. If it might be a headset issue, maybe if it continues, you can write your question in the chat.
Okay, you can hear me now?
Oh, yeah, perfect now.
Okay, no, that's good because I talk after. That should not be nice to go without. Yeah, just a short question. In terms of morphology and or maybe growth as well, the microbiome has been described to be quite involved. So I'm just wondering if when you look at these giant mutants, I mean the different strains that you assess, if you consider some way of normalizing the microbiome across all your individuals, or you just ignore that and go with a local microbiome where you collect it?
Yeah, I mean, that's tricky. Yeah, so it has been like, the microbiome definitely influences the phenotypes, but like we only control, like they're in the same medium, nutrients, light, temperature. So this is what we control, and the rest we don't. So once we sampled, we washed them, and then we put them into bottles, but then the local, microbiome is.
I was just wondering if there was possibility maybe to put them all in the same tank, sharing the same water in some way, that in some way you homogenize and try to clean for the microbiome effect. Just I was wondering if you were doing that.
Yeah, I mean, they are in the same seawater, autoclaved seawater for cultivation. I hope this answered.
Yeah, thanks.
Okay. How are we doing? Let's see what time is now. Actually, we're slightly over time, so maybe we might just continue with Ronald's presentation, if that's okay. And any further messages can be put in the chat. And I think Ronald is from University of Galway and is part of the SeaMark project. And again, would like to talk about Ulva.
Yeah, thank you, Mev. So maybe next slide. Let's go on. We are late. So just to a little summary about in terms of biology, what is necessary in farms if you want to breed efficiently Ulva and grow it. So the first point is probably biobanking, which is a major issue with seaweeds, difficult to maintain them. So there we developed a cryo preservation method. The second part is that if you want to breed, you need to control the life cycle, which can be an issue as well. So we talk about that in the next slides. And you need as well to be able to phenotype a lot of strains for low cost that you you can really find the best ones. And I will not talk about that today. Environment factors are also very important. Next slide. So, okay. My slide has been a bit destroyed as well. That's okay. So the phenotype of your ulva in terms of growth capacity and biochemical composition is in fact influenced by several factors. So you have the genotypes, what I will talk about, you have the environment, and you have also the bacteria. And all these three parameters will as well interact, and that will define your phenotype. So how can we play with the genotype? I mean, what is the variation there? Next slide. So that's an experiment which was done at the time of GeneAlg. So 400 individuals were sent to Galway for phenotyping coming from a number of countries. Next slide. So we could actually grow properly 250 of them. So they were grown for at least 3 weeks and often much longer in the same media, light conditions, temperature, and so on. And then we measure their growth rate. So here you have the results for around 250 of them. And you can see that you have a huge variation in terms of growth rate when they grow in exactly the same environment. So it's definitely a genetic effect. And if you look at the proteins, you have exactly the same variation, measure variations. So that actually fully justifies strain selection for farming. And next slide. Yeah, next. And the very important point is that you might think that if you select for growth, you might actually go against quality of your biomass. So it's something that we looked on the left. You have a correlation between protein and growth rate, and there is no correlation. So it's good news because it means that you can select both for growth and protein content and find individuals which will be top quality and top growth. Okay, next slide. So now I would like to talk a little bit about the possibility to go further, selection in the wild. And in our case, we went for mutagenesis. Next slide. Okay, so the first thing that we had a lot of issues with was to induce sporulation. So Compressa is quite well established. We had a lot of problem with La Simulata, and that's the SeaMark project. So there you have a picture showing you sporulation of a tally. Next slide. And we tested basically 6 different protocols, in fact more, but we extensively checked for 6 protocols. And we found that actually cold stress was extremely efficient on the over 3 species on the around 40 different strains. We could actually achieve a success rate of sporulation of around 80%. So that works, and that's good news. So now we could go for mutagenesis. So mutagenesis, in our case, we use EMS, so that's random mutations. We can apply it on gametes or spores. We could as well use tally if we want, but we chose to go for gametes or spores. The density and the number of mutation will actually depend a lot on the dose of EMS you apply. Can you put click one more? No. Oh gosh. Yeah. Okay. My slides are destroyed as well. Actually, the interesting point is based on the quantity of EMS and the duration, you can build a kill curve. And if you go for high survival, that means that your mutants will be very low in mutations. But it means that if you want to find your phenotype you want, you will need to screen a lot of gametes, which is not a problem with ulva because you can have millions of gametes. And it makes a huge difference with land crops. So that's definitely an advantage to use this approach in Ulva. Next slide. Okay, so there you have an example in La Simulata of a kill curve. So on the graph, you have 4 different doses of EMS, and based on the survival of your gametes, you can build this kill curve, and then you can actually choose the right concentration and duration of of EMS, which will fit your selection. Next slide. So what we did as well on another project that's not SeaMark anymore, that's Marie Curie postdoc fellowship, and there we are using as well, but this time on compressor, EMS approach to identify genes involved in nitrogen metabolism. So the idea is to mutagenize, but then go for on an additional selection, which is about using chemicals which will kill the mutants which are not mutated in our genes of interest. So the nitrogen pathway, basically. Next slide. So there you have the picture on the left, the first mutants that we actually could isolate after this screen. And now we are trying to characterize them and try to identify the genes. Next slide. Yeah, and that's it. So the work presented has been about a number of people, a lot of external collaborators that I did not mention, but there has been a lot, like Roscoe, for example. And most of the work on mutagenesis has been done by Dr. Farhan Habibi and Serena Rossignoli. Thank you.
Thank you very much, Rona. And again, apologies for the little bit of messing up of the slides. And if the presenters are agreeable, we may share the slides. We'll ask your permission afterwards, but it might be nice to share the slides with everybody in the audience, so they can see completely what you meant. So does anybody have any questions for Rona? Not so far. Rona, have you upscaled any of this work into larger biomass trials, or is this all done in your lab?
No, for the mutants, no. We are still screening the mutants. For SeaMark project, we are still in the selection process. So we have a lot of mutants, and now we are making garden experiment, growing them all together, and progressively we expect that the best performers will take over, and then they can be checked at larger scale. For the strain selection, yes, we have been selecting strains, and they have been tested in Portugal.
Okay. Cicero has sent a chat message in here to Rona. Have you measured any correlation of growth rates in gametophytes and sporophytes? Also juvenile, I'm not sure if that's juveniles by adults or if it's, yes, I'm not quite sure.
That's, if I understand properly, is about looking if gametophytes have different growth rates than sporophytes. No, that's actually something we really wonder as well about. I mean, there is all this idea of ploidy, polyploidy, increasing growth rates. I mean, it's sometimes yes, sometimes no. But if it is true in Ulva, you would expect gametophytes to grow less fast than sporophytes. And I am not sure it has been really shown up to now. And that's something which we should look at definitely, because already that will answer part of question about ploidy levels.
That makes sense.
So the answer is no, but I am planning to take a look at that. Yes.
Great. Any other hands that we've missed? No. It sounds like Ulva is actually a little bit quicker to produce different generations than Saccharina, perhaps, which is nice. But I don't know, maybe I'm dreaming.
No, it's faster, I think. I agree. Yeah.
Oh well, we'll not think about it too much. Okay, so if we don't have any more questions for the presenters at this time, of course, ask them anytime. But what we're going to do now is take a 5-minute break, so you can all run off and get the kettle on, and then so that gives us time to organize everybody into the breakout rooms. So there's actually 77 people here on the workshop, which is great. So that's about 12 to 13 people per group. And each group is going to be led by a moderator who will ask the questions, and you will also have an opportunity to answer the questions on your computer as well. We hope that you do. So there will be a link to the webpage for the questions, or there's a QR code. There's a couple of different ways you can do it, but your moderator will explain all of that. So yes, thank you very much for listening. Thank you to our 4 presenters for some really interesting talks. And yeah, let's head to the breakout rooms, Maya. Are we all back?
Yes.
I'm there. So I don't know what happened.
That was we were just getting into a nice conversation, so I don't think we've actually even finished all our questions.
But I see now different people in the room. Did we move to another room? I think.
We were
No, I think we've this is the full. We're all back in all the rooms have been brought back together again,
Okay,
Job. So
yeah, that happened.
Yes. Okay. So well, I failed as a moderator to keep everything on time, but the chats were good. So that was great.
There's only 45 people. I don't know if that's normal.
I don't know. Maya, do you know how many do people leave the breakout sessions and not come back?
I think so, because when we left for the break, it was not as many as in the beginning. So
okay.
So probably not all wanted to be part of the discussions.
Yeah, well, that is fair. So this is the dedicated group now. That's not too bad. 45 is pretty good. Okay, so we have 6 moderators, and Erd, would you like to go first? I'm just picking people at random, but just if you want to give a quick overview.
I can.
Yeah.
How long time do you want us to spend? 1 minute or something like that?
1 or 2 minutes, but just briefly to say to everybody, there were the six groups were divided into 2 sets of questions. So one group will had you know, where it's kind of where do we see the innovation of these technologies going, and then group 2, which you and I were in, was more to do with challenges and barriers surrounding that development as well.
Yeah, exactly. So already from one of the first questions, we talked about the risks and the benefits, that it's very much needed, but everyone wants to be responsible, and Gabrielle also shared a publication that the EU industry or the seaweed industry in Europe today is very responsible. There's not a lot of different foreign strains placed at at other locations. But it was also mentioned by Seaweed Solution that we need some guidelines, and the group, none of us was aware of any law or regulations from neither Europe or national levels. So the conclusion was very much that we should not do the same mistakes that was done in Asia. We can get inspiration from land, where they are now changing a bit back, maybe allowing GMO, since the techniques are very similar when you select for something. But at sea, the ocean environment is one big pool almost, so we need to be careful, and therefore we were all like, we want the regulations, we want the guidelines, the rules, but in a meaningful way where it's not stopping the industry from scaling, but it is helping to make responsible businesses.
Thank you very much, Erd. And actually, seeing as we're talking about this set of questions, I'll continue on with mine because they're of a similar, and then I think it was Lean. I have to see whoever had the third group 2 can pop in after me. Yeah, I think we sort of came to similar conclusions, although we were warned to maybe learn from land crops that do have quite a reduced genotype for certain species now, and to learn from that and try and not let that happen. Possibly areas of protected wild populations that cultivation doesn't exist around, but it really, obviously the scale matters, and when it's on a small scale with the simpler strain selection work, it's maybe not such a big deal, but obviously as technologies and geography scales increase, then obviously that has to be revisited. We were also I think we were mainly a group of scientists in our group, but we weren't aware of European regulations around in this space. So I find that very telling as well. I think this needs to be at least a review of what's out there needs to be done. And regulations weren't well known. There was talk of the 100-kilometer rule, which actually came from Hortimara in the first place some time ago. But there are loopholes to that that can be found. But really, everybody was quite agreed that regulation is required. Scientific data, it should be based on scientific data, but we may need to have more data of this to actually make it species specific, and but at the moment keep it precautionary until we understand more. So basically, it's a lot more modelling. Sounds like it needs to inform the legislation. Okay, who had group 2? Who was the final one for group 2, moderators? If you can just sing out, that would be great.
I don't know the number of the group, but okay.
We have
sorry,
the
yes.
group 2 questions. Yeah. If you are looking for the group 2 questions, are you?
Yes, group 2 questions. Yeah.
Okay, so I'll just go ahead. So our group, yeah, we discussed, I think, the risk of spreading and mixing with natural seaweed populations. And I think the main conclusion is that we don't know much to be able to say anything about it. And the same was the case for other, the spread of diseases and nutrient carrying capacity, that we don't know much. And then we discussed regulations. So there are actually in Norway, there is some regulation, there is a law, but it's difficult to know how to interpret it. And we discussed that there are some regulations that are on how to cultivate. So in France, for example, the cultivators of Undaria, they must harvest before the reproductive development. Things like this is also relevant. And then we discussed population genetics and the population genetic studies. It's how can they be used to give us scientific reasons for how to regulate the business. It's not so easy to transfer that knowledge or how to actually use that information. Okay, sorry, I think that's all from my group.
Thank you, Asheld. We'll move quickly on to group 1. So Lean, do you want to lead with that?
Okay, yes. So in the group 1 questions, in my group we had a mixture of researchers and industry, which was quite interesting. And the first discussion we had was on biomass and how biomass could be improved by breeding. And actually the outcome of the discussion was that in the group it was thought that other things like production systems and so on will have a bigger impact at the moment on the biomass production than breeding would have. We also discussed a little bit on things that need to be improved, and we think also that there a framework, it was only touched for a small part, but that the framework is needed and regulation, so that it's not only about ecology and economy, but also the politics need to be, to set out some guidelines. And then also in the discussion, because we were asked some specific questions on biochemicals, on how we think the production should improve and so on. There the outcome is actually that we think that many things will be market driven. So it will be in the end the market that will also have an impact on how things will be upscaled or what specific compounds will be needed. So that was also the outcome on that type of question.
Thank you very much. That's brilliant. So I'm not sure, Susan and Aoife, I'm not sure if you're in again.
Yep, we're here.
Yeah, oh, brilliant. Yeah.
So,
I can't see you on my screen,
right,
but that's great. If you want to make some conclusions, that would be great.
Yeah, so our group, we had a mixture of researchers and industry as well, and for ideas to improve production, it was highlighted that both breeding and farm design need to improve simultaneously. So there's still work to make sure that without breeding, you still have your lines at the correct depth and all the logistical things are in order, as well as improving on the breeding. And then when we're looking at the market and what would be realistic for a harvest, doubling in 10 years would be very good. And also based on the market, it would tell us what compounds we need. So Signa mentioned that in a project that they had improved the stipe length of Saccharina, so that improved alginate. So that would be good for that industry. And then looking at what does the successful use of breeding technologies within the macroalga sector look like? We were saying if you were to use strains across geographical areas, they would have to be sterile. A European biobank is a priority, and it should be open access.
And then we had some questions about what other desirable traits there were and what were most important, and some of them were fertility. So for Ulva, a desirable trait would be for fertility, and for the likes of kelp, it would be epiphyte resistance to help the crop grow a bit longer. And then we were asked what other than sugar kelp and Ulva, what's the next species to target, and we said Palmaria, Paphyra, and there's one other one, Fusilaria as well. So they were the 3 species that came up that people were interested in from our group.
Brilliant. Thank you very much. And Sophia is the last group. I'm trotting on here because we're a little bit over time, but
Yeah.
it is all very interesting. So
I'm going to be quick. So we had a nice conversation. Thank you for that. Globally on how the strain should be restricted to area of basin, we were in agreement that we need to have region-specific strains or at least basin-specific strain. We have identified some need from research, so the defining of region for each species that could be different, but also some research on how the farm could influence the wild population, so the flow between the farms and the natural population. We then spoke about how much should be the improvement in yield, and the fact is more than the yield that should be improved, it's the reliability and the continuity of the yield. So we need some strains that produce not obligatory more, but at least on a constant manner. We also finished on the problem that we should put the effort of breeding on species for which there is an actual market where you can sell your algae. It's apparently not the case for Saccharina yet. And that the breeding and the harvest improvement should be accompanied by the development of product from these species to create the market and to be able to sell the algae that we produce. And we've been cut on this, I think. That's it for our group.
that's it from your group. Brilliant. Okay, well, thank you everybody for these thoughts. And what I might do is to actually send a follow-up with presenter slides and a link to the recording, and also maybe a link to those questions, so that if anybody didn't have a chance to record them while the chat was going on, that you can do so in your own more relaxed time. But it seems like we have an awful lot more to do, and it looks like we also have a lot of experience across Europe, but that perhaps it should be a bit more coordinated in terms of strain selection work and production. So I think this might open up quite a few interesting conversations in the future. And I'm hoping that this would be something that we continue to discuss, not just at this workshop. Does Erd or anybody else, Asheld, is there any other remarks you'd like to make from the project?
No, I think you summarized it very well, and it's very good that the two projects can connect and make such an event together, because it's very complementary with the work that was done in your project and the SeaMark project.
It's actually quite rare, isn't it? We don't, not often it happens, but I'm glad it has. Okay, well, I'm conscious of everybody's time, so thank you very, very much, and I hope you enjoyed it all. And feel free to contact either Blue Bio Boost or SeaMark for if you know us now, so contact us if you need us, and yep, follow along for more results, I guess.
Thanks for joining.
Yeah. Okay.
Yeah, thank you.
Thank you very much. Bye.
Thank you, Maeve.
Thank you very much. Bye.
Bye.
Thank you.
Bye.
Goodbye.